ये सेकंड पार्ट है प्लांट ग्रोथ एंड डेवलपमेंट का। जैसा कि पहले पार्ट में क्या नाम है, यहाँ तक तो कंप्लीट हो गया था क्वांटिटेटिव कंपैरिजन तक, लेकिन उसके आगे का बचा हुआ है, तो ये सेकंड पार्ट में इसकी रीडिंग है। सो लेट्स स्टार्ट। Quantitative comparison between the growth of living system can also be made in two ways. First, measurement and the comparison of total growth per unit time is called the absolute growth rate. Second, the growth of the given system per unit time expressed on a common basis. Example, per unit initial parameter is called the relative growth rate. In figure 15.7, two leaves A and B are drawn that are of different size but shows absolute increase in area in the given time to give leaves A1 and B1. However, one of them shows much higher relative growth rate. Which one and why? 15.15. Condition for growth. Why do you not try to write down what you think are necessary condition for growth? This list may have water, oxygen and nutrients as very essential elements for growth. The plant cells grow in size by cell enlargement, which in turn require water. Turgidity of cells help in extension growth. Thus, plant growth and further development is initially linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps in realizing metabolic energy essential essential for growth activities. Nutrients, macro and micro essential elements are required by plants for the synthesis of protoplasm and act as a source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain stages of growth. 15.2 Differentiation and de-differentiation and redifferentiation. The cells derived from root apical and shoot apical meristems and cambium differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. During differentiation, shell undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a trachea element, the cells would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell walls to carry water to long distance even under extreme extension. Try to correlate the various anatomical features you encounter in plants to the function they perform. Plants show another interesting phenomenon. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. This phenomenon is termed as de-differentiation. For example, for example, formation of meristems, interfascicular cambium, and core cambium form fully differentiated parenchyma cells. While doing so, such meristems tissue are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific function. Example, that is, get redifferentiated. List some of the tissue in a woody dicotyledonous plant that are product of the redifferentiation. How would you describe a tumor? What would you call the parenchyma cell that are made to divide under controlled laboratory condition during plant tissue culture? Recall in section 15.11 we have mentioned that the growth in plants is open, that is, it can be indeterminate and or determinate. Now we may see that even differentiation in plants is open because cells tissue arising out of the same meristems have different structure at maturity. The final structure at maturity of a shell tissue is also determined by the location of the cell within. For example, cells position away from root apical meristem differentiate as root cap cells, while those pushed to the periphery mature as epidermis. Can you add a few more examples of open differentiation correlating the position of a cell to position in an organ? 15.3 Development is a term that includes all changes that an organism goes through during its life cycle, from generation of the seed to senescence. Diagrammatic representation of the sequence of process which constitute the development of a cell of a higher plant is given in figure 15.8. It is also applicable to tissue/organ. Plants follow different pathways in response to environment or phases of life to form different kinds of structure. These abilities called plasticity. Example heterophyly in cotton, coriander, and larkspur. In such plants, the leaves of juvenile plants are differentiated in shape from those in mature plants. On the other hand, differences in shapes of leaves produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. Figure 15.9. This phenomena of heterophyly is an example of plasticity. Thus, growth, differentiation, and development are very closely related events in their life of a plant. Broadly, development is considered as the sum of growth and differentiation. Sorry. Development in plants example, that is, both growth and differentiation is under the control of intrinsic and extrinsic factor. The former include both intracellular genetic or intercellular factor chemicals such as plant growth regulator, while the latter includes light, temperature, water, oxygen, nutrition, etc. 15.4 Plant growth regulator. 15.41 Characteristics. The plant growth regulator PGRs are small simple molecules of diverse chemical composition. They could be indole compound, indole 3 acetic acid IAA, adenine derivative 9,6 Furified furyl aminopurine kinetin, derivative of carotenoid, abscisic acid ABA, terpenes, gibberellic acid, GA3 or gases ethylene C2H4. Plant growth regulator are variously described as plant growth substance, plant hormones or phytohormones in literature. The PGRS can be broadly divided into two groups based on their function in a living plant body. One groups of PGRS are involved in the growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are called plant growth promoter. Example: auxins, gibberellins, and cytokines. Auxin, gibberellins, and cytokines. The PGRS of the other group play an important role in the response to wounds and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the group, but it is largely an inhibitor of growth activity. 15.42 The discovery of plant growth regulator. Interestingly, the discovery of each of the five major groups of PGRS have been accidental. All this started with the observation of Charles Darwin and his son Francis Darwin when they observed that the coleoptile of candy grasses responded to the unilateral illumination by growing towards the light source, that is phototropism. After a series of experiments, it was concluded that the tip of coleoptile was site of transmittable influence that caused the banding of the entire coleoptile, that is given in figure 15.10. Auxin was isolated by F.W. Wendt from tips of coleoptiles of oat seedlings. The Bacne foolish seedling disease of rice seedling was caused by the fungal pathogen Giberella fujicuroi. E. Kurusu 1926 reported the appearance of symptoms of the disease in rice seedlings when they were treated with sterile filtrate of the fungus. The active substance were later identified as gibberellic acid. F. Skoog and his co-workers observed that from the internodal segments of tobacco stems, the callus, a mass of undifferentiated cells, proliferated only if, in addition to auxins, the nutrients medium was supplemented with one of the following extracts of vascular tissue: yeast extract, coconut milk, or DNA. Miller et al. 1955 later identified and crystallized the cytokinesis promoting active substance that they termed kinetin. During mid 1960s, three independent researches reported the purification and chemical characterization of three different kinds of inhibitor: inhibitor B, abscissin second, and dormin. Later, all the three were proved to be chemically identical. It was named abscisic acid. H.S. Cousins in 1910 confirmed the release of volatile substance from the ripened orange that has stemmed the ripening of stored unripened bananas. Later, this volatile substance was identified as ethylene, a gaseous PGR. Let us study some of the physiological effect of these five categories of PGR in the next section. Sorry. 15.43 Physiological effects of plant growth regulator. 15.431 Auxin. Auxin, from Greek auxin, to grow, was first isolated from human urine. The term auxin is applied to the indole 3 acetic acid IAA and to the other natural and synthetic compounds having certain growth regulating properties. They are generally produced by the growing apices of the stems and root, from where they migrate to the region of their action. Auxin like IAA and indole butyric acid IBA have been isolated from plants. NAA, naphthalene acetic acid, and 2,4-D, 2,4-dichlorophenoxyacetic are synthetic auxins. All these auxins have been used extensively in agriculture and horticulture practices. They help to initiate rooting in stem cutting and application widely used for plant propagation. Auxin promote flowering. Example, in pineapples, they help to prevent fruit and leaf drop at early stage, but promote the abscission of older mature leaves and fruits. In most higher plants, the growing apical bud inhibit the growth of lateral axillary buds, a phenomena called apical dominance. Removal of shoot tips, decapitation usually result in the growth of lateral bud that is given in figure 15.11. It is widely applied in tea plantation, hedge making. Can you explain why? Auxins also induce parthenocarpy. Example in tomatoes. These they are widely used as herbicides to 4D widely used to kill dicotyledonous weeds. Does not affect mature monocotyledonous plant. It is used to prepare weed-free lawns by gardeners. Auxins also controls xylem differentiation and helps in cell division. 15.432 Gibberellins. Gibberellins are another kind of promotory PGR. There are more than 100 gibberellins reported from widely differentiated organisms such as fungi and higher plants. They are denoted as GA1, GA2, and GA3, and so on. However, gibberellic acid GA3 was one of the first gibberellins to be discovered and remains the most intensively studied form. All GAS are acidic. They produce a wide range of physiological responses in plants. Their ability to cause an increase in length of axis is used to increase the length of grapes stalks. Gibberellins cause fruits like apple to elongate and improve its shape. They also delay senescence, thus the fruits can be left on the tree longer so as to extend the market period. GA3 is used to speed up the malting process in brewing industry. Sugar cane stores carbohydrate as sugar in their stems. Spraying sugarcane crop with gibberellins increase the length of the stem, thus increasing the yield by as much as 20 tons per acre. Spraying juvenile conifers with a GAS hastens the maturity period, thus leading to early seed production. Gibberellins also promote bolting, internode elongation just prior to flowering in beet, cabbage, and many plants with rosette habit. 15.433 Cytokinins. Cytokinins have a specific effect on cytokinesis and were discovered as carnitine, a modified form of adenine and a purine from the autoclaved herring sperm DNA. Carnitine does not occur naturally in plants. Search for natural substance with cytokinin like activities led to the isolation of zeatin from corn kernels and coconut milk. Since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compound with the cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapid cell division occur. For example, root apices, developing shoots, buds, younger fruits, etc. It helps to produce new leaves, chloroplast in leaves and lateral shoot growth and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote nutrient mobilization which help in the delay of leaf senescence. 15.434 Ethylene. Ethylene is a simple gaseous PGR. It is synthesized in large amounts by tissue undergoing senescence. Sorry. And ripening fruits. Influence of ethylene on plants include horizontal growth of seedlings, swelling of the axis, and apical hook formation in dicot seedlings. Ethylene promotes senescence and abscission of plant organs, especially of leaves and flower. Ethylene is highly effective in fruit ripening. It enhances the respiration of it enhances the respiration rate during ripening of the fruits. These rise in rate of respiration is called respiratory climactic. Okay. Ethylene breaks seeds and bud dormancy, initiate germination in peanut seeds, sprouting of potato tubers. Ethylene promotes rapid internode petiole elongation in deep water rice plants. It helps upper parts of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapple. It also induces flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as source of ethylene is ethaphone. Ethaphone Ethaphone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Ethaphone hastens fruit ripening in tomatoes and apples and accelerates absorption in flower and fruit. Thinning of cotton, cherry, walnut. It promotes female flower in cucumber, thereby increasing the yield. 15.435 Abscisic acid. As mentioned earlier, abscisic acid was discovered for its role in regulating abscission and dormancy, but like other PGRs, it also has other wide ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of a plant metabolism. ABA inhibits seed germination. ABA stimulates the closure of stomata and increase the tolerance of plant to various kinds of stress. Therefore, it is also called the stress hormone. ABA plays an important role in seed development, maturation, and dormancy. By inducing dormancy, ABA helps seed to withstand desiccation and other factors unfavorable for growth. In most situations, ABA acts as an antagonist to GAS. We may summarize that for any and every phase of growth, differentiation and development of plant, one or other PGR has some role to play. Such roles could be complementary to or antagonist. This could be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect that event. Example: dormancy in seeds, birds, abscission, senescence, apical dominance, etc. Remember, the role of PGR is of only one kind of intrinsic control, along with the genomic control and extrinsic factor. They play an important role in the plant growth and development. Many of the extrinsic factors, such as temperature and light, control plant growth and development via PGR. Some of such events could be vernalization, flowering plants, dormancy, seed germination, plant movement, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factor on initiation of flowering. 15.5 Photoperiodism. It has been observed that some plants require a periodic exposure to light to induce flowering. It is also seen that such plants are able to measure the duration of exposure to light. For example, some plants require the exposure to light for a period exceeding a well defined critical duration, while others must be exposed to light for a period less than this critical duration before the flowering is initiated. In them, the former group of plants are called long day plants, while the latter ones are termed as short day plants. The critical duration is different for different plants. There are many plants, however, where there is no such correlation between exposure to light duration and induction of flowering response. Such plants are called a neutral plant, that is given in figure 15.12. It is now also known that not only the duration of light period but that the duration of the dark period is also of equal importance. Hence it can be said that flowering in certain plants depends on not only on a combination of light and dark exposure but also their relative duration. This response of plants to periods of day night is termed as photoperiodism. It is also an interesting to note that the while shoot apexes modify themselves into flowering apexes prior to flowering, they that is Shoot apexes of plants by themselves cannot perceive photoperiods. The site of perception of light dark duration are the leaves. It had been hypothesized that there is a hormonal substance that is responsible for flowering. This hormonal substance migrates from leaving to shoot apexes for inducing flowering only when the plants are exposed to the necessary inductive photoperiod. 15.6 Vernalization. There are plants for which flowering is either quantitatively or qualitatively dependent on exposure to low temperature.